Brains Without Break, a new insight into aging phenomenon. Back in the old days, it was thought that as age advances, brain cells are lost day by day, but studies have disproved that. In fact, the neurons do not die with normal aging. There is a widespread conservation of brain cells as one ages, and they show some changes in the way they communicate, both electrically and chemically. There is a decreased number of synapses, or connections, between cells, and changes occur mainly in the neurotransmitters systems, the chemicals that communicate between cells. The main changes in the signaling pathway within the neuron. Pathways used to promote neural outgrowth and learning are disabled, but the actual survival pathways stay intact. Neuroplasticity is the brain's ability to modify its synaptic connections and function in response to environmental demands. It is an important process in learning. Plasticity in the young brain is very strong as they learn to map their surroundings using the senses. As one grows older, plasticity decreases to stabilize what they have already learned. This stabilization is partly controlled by a neurotransmitter called gamma-aminobutyric acid, GABA, which inhibits neuronal activity. This role of GABA was discovered by K.A.C. Elliott and Ernst Fleury at the Neuro in 1956. Researchers at the Montreal Neurological Institute and Hospital of McGill University examined the effects of aging on neuroplasticity in the primary auditory cortex, the part of the brain that processes auditory information. The study was published in the eNeuro, a journal of Society for Neuroscience, on the 21st of August 2018. Dr. Mike Cisneros Franco, the first author, and Dr. A. Tilda Shienda Villas-Sidani, lab director, tested the hypothesis that plasticity stabilization processes become dysregulated as one ages. They experimented on the neurons in the primary auditory cortex of rats to measure how they adapt their responses to the sound. They found that tone exposure caused neurons in older adult rats to become increasingly sensitized to the frequency, but this did not happen in younger adult rats. The effect in the older adult rats quickly disappeared after exposure, showing that plasticity was indeed dysregulated. However, by increasing the levels of the GABA neurotransmitter in another group of older rats, the exposure-induced plastic changes in the auditory cortex lasted longer. These findings suggest the brain's ability to adapt its functional properties does not disappear as one ages. Rather, they provide evidence that plasticity is, in fact, increased but dysregulated in the aged brain because of reduced GABA levels. Overall, the findings suggest that increasing GABA levels may improve the retention of learning in the aging brain. The study showed that the aging brain is, contrary to common belief, more plastic than the young adult brain. On the flip side, this increased plasticity meant that any changes achieved through stimulation or training were unstable, but both easy to achieve and easy to reverse. However, it is possible to reduce this instability using clinically available drugs. Researchers and clinicians may build upon this knowledge to develop rehabilitation strategies to achieve the full plastic potential of the aging brain. Authors say you can teach old dogs new tricks, but the research shows you can teach an old rat new sounds, even if the lesson doesn't stick very long. Thank you.